please, I ask the speaker to be in time. The first one, Professor Michael. He is from the Institute of Immunology, Hamburg, Germany. He did his veterinary medicine in Germany, and then he did his master and PhD in German cancer research facilities in 82, and he is a professor of immunology and medical faculty, University of Hamburg, and also is having copies of membership. His research activities is very wide. His work in analysis of molecular mechanisms employed by tumors, and also his work in complement in inflammations, that in renal diseases and graft rejection, and also his work in comprehensive complement analysis. He's having a very good number, around more than 42 original publications in international reviewing journals, 43 review books, and some in PubMed. As we know, it is a very, very high journals. Also, he is a reviewer for international journals like Nature and Journal of Immunology and Biochemistry. I am sure we will enjoy his presentation. Please, you have the chair.
But I'm from Heidelberg, Germany. The University of Heidelberg is the oldest university in Germany. We are just currently celebrating our 625th anniversary this last weekend. So this is just for the beginning. And I'm very pleased to be here. The action of complement with respect to tumor defense. And I think if you screen the literature over the last years, you may have noticed or not noticed that actually complement is almost never mentioned in context of tumor immune response. And the question, of course, was why? Because we all know that complement is a very powerful tool. And what you can see here on this first slide, this opening slide, this is actually the view of a very famous German scientist 100 years ago, Paul Ehrlich. And he considered complement as a molecule which connects to an antibody, and the antibody, of course, connects to a pathogen of a microorganism. And he did not know, of course, that this one molecule would turn up 60 to 80 years later to be a system, an entire system of the immune system, the complement system, which more than 35 proteins acting within a cascade reaction as regulators, as receptors on cells. So the question is, how is complement related to tumors? Just for those of you who are not dealing with complement every day, I will give you a very brief, not comprehensive introduction. We consider nowadays the complement system one of the major danger sensing systems. That means they sense dangerous molecules. Of course, primarily the dangerous molecules coming from the pathogen, microbes, microorganisms. But in addition, this extends our view. There are also danger signals coming from apoptotic cells. Apoptotic cells need to be eliminated because they provide a source for autoantigens. and altered cells. And among the altered cells, there may be also tumor cells, and there may be cells after destruction in course of, for example, polytrauma, sepsis, or burn. So what happens? These so-called sensing molecules bind to these cells or to the microorganisms, and then the cascade, an enzymatic cascade reaction is initiated by 3 different pathways, and then this leads to the activation of the central component of complement, which is named C3. You see complement is C, short form, and then it's numbered. And then it continues to the activation of C5, and then a so-called membrane attack complex is generated, which inserts into biomembranes and kills either the pathogens or cells or our own tissue, for example, in cases of autoimmune diseases or immune complex diseases. So once this cascade has occurred, we get many so-called cleavage products, small peptides with biological function. For example, C3b and some other more, and those, of course, interact with complement receptors on different cells. And there's no cell in the body which does not have at least one of these complement receptors. Not only the circulatory cells in the immune system, but also each cell in our tissue. That means this provides the information that complement is very important, not only in the defense of microorganisms, but also to keep homeostasis and to be involved in many processes. What are the biological functions? We all have learned in the textbooks the lysis of microorganisms. But much more important is the activation and the recruitment of the cells to the place of infection. And this leads, complement activation, to the process of opsonization, leading to better phagocytosis of pathogens, and also, which is extremely important, to the regulation of immune complexes. Every day, immune complexes are developed in our body, and they need to be eliminated. If they are not eliminated, they will be trapped in the small vessels of the kidney and the joints and the skin, and severe inflammatory diseases will develop. These immune complexes, after being opsonized with complement C3b, they are carried by erythrocytes, by red blood cells, to the liver and spleen, the so-called reticuloendothelial system. What is equally important is the connection of complement as being a major part of the innate immune system to the adaptive immune system. So complement activation products leads to the regulation of dendritic cells in their antigen presentation. It also impacts on B cells in antigen recognition and in the regulation of T cells. This picture, a sketch showing these Roman guys, indicates the symptoms, the 5 key symptoms of inflammation, as we all have learned in medical school. This is the heat, the redness, the tumor, which is not a tumor as a cancer, of course, which is edema formation. Edema formation is extremely important in our body defense because it brings into the tissue very important proteins and cells. But if it comes to pain, in Latin, these are Latin names here, we all have to learn Latin in Europe for medicine. It comes to pain and to loss of function of the organ, then it becomes very interesting to clinicians. This is a great part of our work doing in my department, dealing with infectious diseases, especially nephritis, angioedema, severe diseases in the complex, and this is a very powerful regulator for some peptides called endotoxins that they get inactivated. Even more, and this is also of high interest with respect to my talk, is that all of our cells, literally all of our cells in our tissue, they are covered with 3 or 4 of complement regulatory proteins. They protect our cells against an unwanted inflammatory reaction. These 3 regulators, CD46, 55, and 35, they prevent the activation of C3. CD59 prevents the insertion of the membrane attack complex in our own tissue. So what's about the resistance of tumor cells against complement? And I will start with a summary slide which we have published, or from data which we have published over the last 10 years. And the tumor cell shows a so-called intrinsic resistance. And this is due to the fact that these membrane regulators, which I have just explained to you, are overexpressed, highly overexpressed on tumor cells. And so you can imagine that if you want to target complement on these tumor cells by means of tumor-specific antibodies, that complement cannot be really activated on these cells. And you all know that meanwhile, at least in several countries of the world, we do have about 10 different so-called humanized monoclonal antibodies which are given as therapy to cancer patients. The 2 probably most well-known and prominent of these antibodies are the so-called rituximab, anti-CD20, to patients who have lymphoma. And the other one is even a little bit older and longer known, is an antibody called Herceptin against an epithelial growth factor on the cells of mammary carcinoma cells. In addition to these membrane regulators which are overexpressed in the tumor cells are able to secrete soluble inhibitors. Usually proteins and also complement inhibitors are synthesized by liver cells, hepatocytes, and in part also by monocytes. But here we could show, and I will show it maybe later if I've included it, that if we treat tumor cells with cytokines, then we get also a release of many of these soluble complement inhibitors into the so-called microenvironment of the tumor. The microenvironment of a tumor is extremely important for the fate of the tumor. The composition of pro-inflammatory peptides of molecules and regulatory proteins, of cytokines and other mediators, they indicate in which direction a tumor will go, either growing or being destroyed. In addition to the soluble inhibitors, there are also enzymes released by tumors which directly inactivate complement components. And these events, the expression of membrane regulators or the release of soluble inhibitors, can be triggered by cytokines, hormones, and drugs. I will give you an example for the cytokines and for drug association and complement resistance. More importantly, probably, sublighting complement, meaning low doses of complement, if they hit the tumor cell gets more resistant to subsequent attack by sufficient complement. I will give you the data of a very interesting experiment there. And then we also see what we call the so-called cross-protection of inflammatory mediators, meaning that if you hit, for example, a tumor cell with a low dose of complement, then it not only gets resistance to complement, but also to perforin, to T-cell cytotoxicity. So this is a summary slide of these 3 pathways again. The classical pathway induced by antibodies. So we take, for example, the situation that a cancer patient gets an anti-tumor antibody, and this cascade reaction is initiated. potentially leading to the lysis of the tumor cell or the opsonization, so the activation of phagocytes and of natural killer cells. But there are also other pathways which may be initiated on the tumor cell. And here you can see where the membrane regulators, where I will focus on in the next minutes, the interfering with the activation at this point here, and the CD59 is interacting with the membrane attack complex. Can I do anything to stop this? No, it stays. There has been an interesting study, and this is just one example of many studies, indicating that this overexpression of complement regulators on the surface does really have an impact on the survival of patients. And this study done by Watson in 2006 indicates that tumor cells which overexpress CD59, that this is associated with a reduced survival in colorectal cancer patients. And this, I hope you can see it from the far. This is the total patient group, and this then now is discriminated in stages. And as you can clearly see, those patients who have in their biopsies of the tumors a low or non-expression of CD59, as indicated here on the right-hand side, they survive much better than those who have a strong expression, an over-expression of this regulatory protein. And if you look for patients which are in the stage 1 and 2, so in the lower stage of the disease, which is still to be continued, this is even more obvious here. This is highly significant. So coming to our experiments, which we did to prove that tumor cells are really complement resistant and that this is under the influence of cytokines, which was done here. So we took breast carcinoma cells and we incubated them with cytokines like IL-4, tumor necrosis factor alpha, and IL-1 beta. And then after this treatment and of course control, and then we exposed these cells to a specific anti-tumor antibody and complement. Normal human serum as a source of complement. Normal human serum, NH8. And then we did the cytotoxicity assay. And what you see here is specific lysis. In buffer control, you see that they have lysis which are 80 to 100% probably, but upon treatment with these cytokines, the lysis reduced, that means the resistance increases. And especially here with tumor necrosis factor, extremely down-regulated the susceptibility of the tumor cells to complement lysis. And if we looked closer, if it is associated with some other events, we looked for the expression on these tumor cells of the complement regulators, not only CD54, 9, and 55. And this really indicates, as we know from other experiments, the extreme variety in response of tumor cells. Although we also see a resistance induction here, this in this case is not correlating with the expression of CD59 or other proteins. Whereas in tumor necrosis factor alpha we have a strong increase of CD59, and in IL-1 beta we even have an expression increase in both regulators, and we did much more experiments, but they all showed quite different pictures. But the bottom line is that we can, and this will reflect the microenvironment situation, that mediators may have an impact on the resistance of complement. This is a summary slide. Actually, behind this is 2 years' work of one of my PhD fellows. We were interested in the fact how drug-resistant cell may respond to complement. We all know that multi-drug resistance or drug resistance is a great problem in clinic. So we had the hope initially that these cells which are drug resistant may be vulnerable to complement and tumor-specific antibodies. But what we found was the following. I will explain the experiment. We used cells, ovarian tumor cells, which were not drug resistant, and we took part of these tumor cells and incubated them over weeks with increasing doses of a drug, doxorubicin, for example. And then we looked if they were drug resistant. There are tests to do to prove that these cells are drug resistant, and this correlates with the appearance of a so-called drug pump, which is called P-glycoprotein. P-glycoprotein pumps out all drugs which are going into the tumor cell. So by this, the tumor cell gets rid of all drugs and is resistant to the drug. And what we see here in this summary slide is that if we treat the non-resistant cells as complement, they can be lysed by a specific antibody as complement. But if they are getting resistant to doxorubicin, as indicated with the blue bar, they lose their susceptibility, they get very much resistant to complement and drug. And then we thought it may be correlating with the appearance of this pump, with this P-glycoprotein. And so we blocked it with cyclosporin A, which is given here in this bar, or with a specific antibody, anti-P-glycoprotein. This is due to the incompatibility of the computers. I have a Macintosh. This is the antibody, and this is the drug. And then we also had a culture where we we took off the drug for a couple of weeks, so they lost their drug resistance. And if we treated these so-called revertants with antibody and complement, they got sensible again. We could lose them, lyse them. So it was connected to the drug resistance that complement could not kill these cells. And if we look closer to the cells, indeed we saw what we almost expected. These are the non-treated cells with a certain level of complement regulator 59, 55, 46. And this is indicating that we indeed had multi-drug resistance because it's the glycoprotein expression. And you see that upon action of this drug on the tumor cell, we got a significant increase of this regulator, which was partly or more significantly reduced after taking off the drug from the cell culture. So what we could conclude that this is not correlating to the appearance of P-glycoprotein, but it is due to the fact that certain drugs are able to increase the regulatory potential on the surface of tumor cells. Another series of experiments, which was previously done in the laboratory of one of my collaborator, and then later continued in my laboratory, was the following. We took cells, erythroleukemic cells, K562. We incubated those with a low dose of complement, which is not able to induce more than 5% killing, and then washed the cells, and then we incubated them with a high dose of complement and specific antibody, or with perforin. Perforin is the pore-forming molecule derived from T cells and NK cells in B cell-mediated killing. What you see here on the left-hand side is that this is without pretreatment, we get a lysis of 80%. If we pre-treat with low dose of antibody and complement, we get a significant reduction of lysis, meaning an increase of resistance. Interestingly, if we incubate those cells which are pre-treated with low dose complement with perforin, we got the same resistance. If we did the experimental round without perforin in the first step, but here perforin pre-incubation, low dose, and then adding perforin in high dose, we got an extreme increase in resistance. Not as extreme, but also very significant, we also got an increase in resistance after the treatment with high dose of complement. This means, in consequence, and this is actually bad news, if the tumor cell is not hit with a high concentration of either complement or a high concentration of perforin from the cytotoxic response of T cells or NK cells, it gets more resistance to the subsequent interaction of either the complement system or the T cells with the tumor cell. So after having, and we did much more work, and if you are interested you can read it in the literature, what should we do? We are facing the situation that complement resistance is a fact in tumors, and this is the greatest barrier of a successful treatment with tumor-specific antibodies. And so the approaches we did is using antibodies to block these regulatory proteins which are on the surface of the cell, or later on we used so-called antisense strategies to down to knock down the expression of regulators. I will give you some examples. First, here using combined anti-breast carcinoma antibody and antibodies to CD59, 55, 46, and a mixture of all these antibodies, and then of course serum as the source of complement. And what you can see here, without pre-blocking of these complement regulators, we see just 10% lysis, even in two different cell lines of breast carcinoma BT474, T47T. If we block just the CD55, we got a nice increase. If we block individually CD59, it's even better in both cells. This is not so expressed here. But if we use all antibodies to block all of these three regulators, we got a very nice killing of the tumor cells, indicating the importance of this regulatory protein. And this was confirmed by an elegant study done by Professor Tedesco in Trieste, a collaborator of mine in these studies. He did a mouse experiment where he targeted these two regulators, CD55 and CD59, on human lymphoma cells which were injected into SCID mice. These are mice which are deficient in their immune system, so they cannot reject the human tumor. And what he did is the following. He had a couple of groups of these mice, non-treated, and those who are treated with an antibody which is now in the clinic, anti-CD20 to lymphoma cells, but this was biotinylated. And then another group was treated with a construct of an antibody which is directed against two of the regulators, 55 and 59, also biotinylated. And you see already many mouse die without treatment. Almost 30% survive with the treatment of rituximab. Actually, this is the treatment of the patients nowadays. And this is even better just blocking the complement regulators. But if you now combine the treatment of rituximab and the construct against the two regulators, and this was a very elegant study, due to the fact that he biotinylated, he used avidine, and you know biotin and avidine, they have a very strong affinity, so he could connect both antibodies to the tumor cell. And then you see they got really a nice survival of 75%. After having done these antibody studies, we thought there may be another way, maybe more efficient way, to prevent the expression of these regulatory proteins and thereby to reduce the resistance of the tumor cells. We use the so-called antisense strategies, and for those of you who are not familiar with this, we used 20 nucleotide double-strand DNA. This is indicated in green, and this given to the cells, and this targets the specific messenger RNA for our regulatory proteins. And by this it's most often an enzymatic cleavage, we could cleave this messenger RNA, and there's no translation, and the expression of the regulators is reduced. And this you can see here in a FACS analysis of the expression of 55 and 46. But these are all controls. We have to use our missense controls. And here we get a reduction of almost 70%, and this is even a little bit higher if we use other cells. Or if we use, let's say, as a target, another complement regulator. What is the consequence? This is the lysis of the resistant tumor cells, and upon the reduction, knocking down, we get a very nice lysis of these tumor cells if we combine the silencing of 55 and 46 regulator. And this also induces a high increase of the so-called opsonine, C3b, on the tumor cell, that not only lysis will occur, but also the activation of NK cells and phagocytes.